Assalamualaikum and good morning everyone. So today we're going to discuss on the factors that affecting the enzyme activity. Okay, and I'm sure some of you, most of you actually, you have learned this during your secondary learning, right? Secondary education, which was during your Form 4 or Form 5, right? Somewhere in between there. Right, so let's take a look. The first factor, which is temperature. Okay, so when it comes to temperature, when we plot rate of reaction or velocity against the temperature, this is what we can expect from the graph. All right, so now let's continue. Now at low temperature, which is at this point, all right, this starting point of the graph at low temperature, right, or at lower temperature, so kinetic energy of the enzymes and substrates are low. Okay, so now class, you need to know that both enzyme and substrate, they have their own kinetic energy. Okay, they have their own kinetic energy. So at low temperature or at low temperature, so both parties, the enzymes and the substrates, their kinetic energy are low. Okay, so enzyme and substrate, they move slow. Okay, when they move slow, so less colliding with each other, so rate of reaction or reaction rate is low. Okay, you can see from this graph the rate of reaction is low as well. Okay, now when we increase the temperature, okay, so as the temperature increases, what happens? Obviously kinetic energy of the enzymes and substrates increases. Betul kan? So when this enzyme and substrates kinetic energy increases, both enzyme and substrate will move faster. When they move faster, frequent colliding with each other will occurs. Okay, so more chances of colliding between enzyme and substrates will occur. So when enzyme collide with substrate, meaning to say the enzyme will bind with the substrate, or the substrate will bind with the enzyme at the enzyme's active sites. When that happens, more enzyme substrate complex is formed. So when we have more enzyme substrate complex, the rate of reaction rate or velocity also increases. Okay, that's why the velocity increases here when the temperature is being increased. Okay, so when the temperature increase, the velocity also increases, right? Because frequent colliding between enzyme and substrate, thus more enzyme substrate complex is being formed. Now the temperature coefficient at this point is 2, right? So if you're wondering what is temperature coefficient, all right, temperature coefficient, all right, is given as rate of reaction at x plus 10 degrees Celsius, okay, divided with the rate of reaction at x degree Celsius. So this temperature coefficient you will learn in detail during your experiment, okay, your experiment later on in the lab. So now, for now, you need to know that for temperature coefficient, it is given as rate of reaction at x plus 10 degrees Celsius divided by rate of reaction at x degree Celsius. We take one example. What is the temperature coefficient at 20 degrees Celsius, for example? Okay, what is the temperature coefficient at 20 degrees Celsius? So the calculation is the temperature coefficient at 20 degrees Celsius is equivalent to rate of reaction at 20 plus 10, which is 30 degrees Celsius, divided with rate of reaction at 20 degrees Celsius. So mean to say 30 divided by 20. So berapa tu? Okay, itu jawapan dia. And when it comes to temperature coefficient, dia tidak SI unit sebab kamu selalu cancel off degree Celsius ni. You already cancel off the degree Celsius. So it's just either a single or double digit here. So better definition of temperature coefficient is, okay, it is the relative change of a physical property when the temperature is changed by 1 Kelvin. Okay. Now at the optimum temperature, which is at the point here, the inflection point, okay, at the peak of this graph, what happens here is vibration of the enzyme and substrate reaches its limit. Okay, the vibration of the enzyme and substrate reaches its limit, because enzyme and substrate, when they have kinetic energy, all right, but there is a limit on how this enzyme and substrate molecules will vibrate due to the presence of their kinetic energy. Okay, no matter how much kinetic energy you supply or temperature you supply to this enzyme or substrate, this enzyme and substrate's vibration, it has its limit, right? So now at this optimum temperature, both enzyme and substrate vibration reaches its limit. So all the enzyme form enzyme substrate complex with the substrate, and then the reaction rate is maximum. Isn't it obvious? It's obvious. Now, above the optimum temperature, what happens when we further increase the temperature above the optimum temperature? So you can expect to get this D part of the graph, right? So the kinetic energy of the enzymes and substrate will further increases. Okay, will further increases. More collision between enzyme and substrate. Enzyme molecules continue to vibrate until the hydrogen bonds as well as ionic bonds that maintain the enzyme 3D structure breaks up. Okay, when this happens, the configuration of the enzyme is changed, meaning to say the active sites no longer fit the substrate. That's why you have here, okay, this is the actual rate of reaction. Okay, so meaning to say this is the part where the kinetic energy of the enzyme and substrate further increases and more collision between enzyme and substrate. But class, all right, why does the formation of the product or the formation of enzyme substrate complex decreases here? Because the concentration of the substrate becomes the limiting factor. Okay, otherwise if the concentration of the substrate is not the limiting factor, this will further go up. Okay, but in this case, substrate concentration is being the limiting factor, so the graph slightly goes down, okay, or fall. The graph slightly fall. Okay, but the graph, what happens when the temperature further increases, the graph continues to fall because the configuration of the enzyme is changed. Okay, and when configuration of the enzyme is changed, the denaturation of enzyme starts. Now, when the enzyme starts to denature, right, the rate of reaction decreases. Okay, now at higher temperature, okay, at higher temperature, meaning to say above the E just now. Right now we are at E. So at high temperature, which is represented with E in this graph, okay, the enzyme already denatured. So there is no enzymatic activities. Now, the conclusion on effects of temperature on the enzyme activity. Okay, rate of an enzymatic reaction increases with increasing the temperature because substrates collide with active sites of the enzymes more frequently. Okay, when both molecules move rapidly. Next, each enzyme has an optimal temperature at which the enzyme rate of reaction is the greatest. Without denaturing the enzyme, this optimal temperature allows the greatest number of molecular collisions between the enzyme and the substrate. as well as the fastest conversion of the reactants to product molecules. Reactants here referring to the substrates. And then above an enzyme optimal temperature, right, above the enzyme optimal temperature, the speed of the enzymatic reaction will drop sharply, as you can see from this graph here. Okay, above this optimal temperature, the rate of the reaction for the enzyme will fall. Okay, the thermal agitation of the enzyme molecule disrupts the enzyme's hydrogen bonds, ionic bonds, and other weak interactions that stabilize the active shape of the enzyme. So the enzyme protein molecule eventually denatures. Most human enzymes have optimal temperature of about 35 to 40 degrees Celsius, which is very close to our body temperature, right? Thermophilic bacteria, all right, that live in hot springs. These bacteria, apparently they have enzymes that has optimal temperature of 70 degrees Celsius and higher. That explains why these thermophilic bacteria, they live in hot springs. And this is how they look like, this tiny thing here, right? So this thermophilic bacteria is a rod-shaped bacteria. Okay, so in this diagram here you can see how the active site of an enzyme change shape, right? And this cause the enzyme cannot fit this cause the enzyme active site cannot fit with the substrate when this enzyme is denatured. All right, now the second factor. All right, so we already talked about temperature. Now second factor is pH. Okay, most enzyme has optimum pH between 6 and 8. Okay, and before I proceed, you know that enzyme and substrates, okay, on the surface of these molecules they have hydrogen bonds and other weak interactions on their surface, right? So now there are exceptions such as pepsin, all right, like what I mentioned earlier. Most enzyme has optimum pH between 6 and 8, but some enzymes like pepsin, okay, works best at pH 2, right? Pepsin, this pepsin enzyme really like acidic environment, very acidic environment. Okay, and then trypsin has an optimal pH of 8. Okay, because these these two enzymes, the pepsin and trypsin, these enzymes are able to maintain their 3D shape in such extreme pH environment. Now, the optimum pH, right, is the most favorable environment for the enzyme to be active. That's why enzymes, they work best in their optimum pH, right? Because in their optimum pH, it is the environment where these enzymes can be active, most active. Now, when pH is being lowered, right, meaning to say further lower than 6, like 5, 4, or 3, right, what happens? The concentration of hydrogen ions increases, right? More acidic solution or more acidic environment has more hydrogen ions or protons. So this high number of hydrogen ions in the environment will disrupt the ionic bonds in the enzyme active sites. Okay, so I need to add here in the enzyme active sites. Okay, so when the ionic bonds in the enzyme active site is already disrupted, okay, what will happen to the enzyme active site? Obviously it will change, okay, and this will ultimately cause the enzyme to not functioning, right? Now what happens when the pH is increases? So when pH is increases, meaning to say we have more hydroxide ions, so the number or the concentration of hydroxide ions, all right, in that environment increases, right? when this high number of hydroxide ions increases, what happens? These hydroxide ions will disrupt the ionic bonds in the enzyme active sites, and ultimately this will change the enzyme active site and cause the enzyme to not functioning. Okay, so very similar explanation when the pH is being lowered. Now, extreme change in pH will cause the enzyme to denature. Okay, so restoring the pH back to its optimum pH usually restores the enzyme. Okay, so meaning to say the effects of pH on enzyme is usually reversible. Right, effects of pH on enzyme is usually reversible. So there is a list of enzymes here, right, with their optimum pH. Okay. Now, C, the third factor, right, is the concentration of the enzyme, right? So at lower enzyme concentration, you can see here when we plot the graph rate of reaction against the enzyme concentration. So at lower enzyme concentration, the rate of reaction is proportional to the enzyme concentration. So I mean to say, as we increases the enzyme concentration, the velocity also increases. Okay, boleh nampak ah? Sini graf ni. Bila enzyme concentration bertambah, rate of reaction also increases. At higher substrate concentration, okay, at higher substrate concentration, this is what we will get. The rate of reaction is constant, right? Velocity is constant. Why? Because all the substrate are in the form of enzyme substrate complex. Okay, so the substrate concentration becomes the limiting factor, and a hyperbola curve is obtained when rate of reaction is plotted against the enzyme concentration. To increase the rate of reaction, what do we do here, right? Since the substrate concentration, right, the substrate concentration becomes the limiting factor, we increase the substrate concentration, right? So to increase the rate of reaction, we increase the substrate concentration so that more substrate are in the form of enzyme substrate complex. All right, now the fourth factor is the substrate concentration. Okay, so now as soon as the reaction starts, the substrate concentration will fall while the product concentration increases. Okay, so the rate of reaction or velocity is proportional to the substrate concentration. Okay, so you can see as we increase the substrate concentration, velocity also increases. Okay, but at one point there is no point on further increases the substrate concentration because the enzyme concentration becomes the limiting factor. So a rectangular hyperbola curve or a hyperbola curve is obtained when we plot velocity against substrate concentration because the enzyme concentration becomes the limiting factor. So we have a constant part of the graph here, right? A constant rate of reaction in the hyperbola curve here. This is because of the enzyme concentration being the limiting factor. Now, further increase in the substrate concentration will cause the velocity to becomes maximum because all the enzyme molecules are saturated with the substrate molecules, or all the enzymes are in the form of enzyme substrate complex. So like what I mentioned earlier, the enzyme concentration becomes the limiting factor. Okay, so that's it for today. Okay, so we already discussed the four factors that are affecting the enzyme activity. Okay, see you in the next class. Bye.